We've had Alistair on for 2 previous webcasts and live in person. I think you were in person, or were you on our live? Yeah, you were, and he's nodding to me, at the EGA Garden States event, and has been progressing with his research and hearing more about it each time. So I'll welcome now Alistair with the sub update. Welcome.
Thank you, Nick. You're warm. Your voice is just so warm. I'm Alistair. I've been working on magic mushrooms for the last 2 years. It's just been a breath of fresh air from my old work or normal work on plant pathogenic fungi. And the work on mushrooms that I've been doing is done on the smell of an oily rag. So I need to thank, or I want to thank, everyone who's helped me along the way, particularly the landowners and the people who've been out there collecting and sending me more than 250 spore prints just last year. Tonight, you will see the hard work of people like Kane, Nigel, Dave H, Jan, Snoo, Bill, and Tim S, who've been sending me subs from all around Australia. Just so helpful. Bioplatforms Australia, they've added some oil to that not very oily rag. They helped sequence 85 genomes of Psilocybe subaeruginosa, which I got back just one day before Christmas. They've just released a new funding announcement. It's called the Fungal Genomics Initiative. If you work in the mycology space and you have a want to sequence a proteome, metabolome, transcriptome, genome of any fungus, then check it out. They could be able to support what you want to do. I'd also just like to acknowledge my co-authors, Tim, Jason, Kevin, and Steve. I like to think of myself as a fungal geneticist, but more as a tourist. I go around and maybe I send you a postcard from where I'm exploring, whereas someone like Tim, he lives there and breathes genetics, and he's like the QC for the quality control for whatever I'm doing. So let's get to it. Kane already touched on a lot of this. Gosh, this is like a little deja vu of what he was doing. I just want to really give you a little bit of background about the history of subs, or maybe more about how people choose to name fungi. So mycologists or taxonomists, they have to do justice to biodiversity. There's a fine line between describing too many names for a particular fungus or too few names and not doing justice to biodiversity. So too many names, you kind of start to lose the power of a name, and every new population becomes a new name. Whereas when you don't have enough species, then you're missing out on diversity. So people, when they look at mushrooms, they're microorganisms. They don't have that many characters to go from. And the reason that mycologists will start to describe new species is based on morphology. So these subs over here, we've got a typical flying saucer mushroom, a nice wavy cap. And then the ones that we've seen lovely photos of, similar to Tanner's photos that were in Kane's talk, that we know and love. So people will see differences and they'll describe a new species of mushroom. So phenotypic differences or ecological differences, fruiting time, substrate. They'll describe new mushrooms when they appear in new places, and maybe they'll go, okay, well, this deserves a new name. And right now, the gold standard for applying a new name in taxonomy is a phylogenetic species hypothesis or phylogenetic species concept. This is just based on 1 or 2 genes, 1 to more genes. It can be lots and lots of genes. And you're essentially comparing your taxon in question, its evolutionary relationships based on a phylogenetic hypothesis, and seeing if it is one thing or another thing that is comparable in its sequence data. And we'll have an example of that later on if you have no idea what I'm talking about. So yeah, there's a little bit of excuses in the history of mushrooms that look like Psilocybe subaeruginosa, where people have thought, well, hey, we should slap a new name on this thing. So when it's been introduced internationally, and we've ended up with a handful of names like cyanescens and azurescens and allenii, that have been named from the Northern Hemisphere that look suspiciously similar to Psilocybe subaeruginosa, or people have even split within Australia and New Zealand applied new names. And then we've had new names applied also based on sequence differences in the ITS region. So some of these more recent names are based on differences between ribosomal DNA genes. So the genes that encode the structure of the ribosome. So with all of the sub data that we have, which is about 85 genomes or 86 genomes, no, it's more than 90 genomes, we can start to test hypotheses. And the hypothesis that I'm testing is that Australia is the center of origin of these wood-loving magic mushrooms. And if you've been to one of my talks before or seen it, you will have seen this funky diagram. This is a SplitsTree. It's actually a SplitsTree of a psilocybe, the relationships among Psilocybe cubensis. And it's just here so that we can practice for when we see the real deal on subs. So a SplitsTree is just a way to visualize the relationships among genomes. And in this case, it's 124 genomes of Psilocybe cubensis, sequenced from around the world and in Australia. The little circles at the ends of these edges or branches are actual genomes. And the difference or the branch length that connects them to things, that is informative of how different they are. So that is indicative of genetic difference. So this network is based on 250,000 differences across a genome. So you can see something that I've never kind of explained in any of my talks is that things like this little 4 that I'm kind of circling with my mouse, I hope you can see it, is those are siblings of Psilocybe cubensis that has been printed, and then I've taken spores from the same mushroom and grown up cultures. Those are siblings. So you can see that even from one mushroom cap or pileus, thank you, Kane, for that beautiful word, you've got these siblings that are still quite genetically different, which is an indication that of heterozygosity in our Australian genomes, especially when you compare them to this pink color over here, which is the golden teacher kind of population, or that I've named the golden teacher population. And I can't see because I've got a big long list of big thing of faces down my screen, but there's golden teacher, B positive, Maui, McKenna, all these popular names are essentially the same thing genetically. There's very little genetic diversity among them. So even less genetic diversity among all these strains than among siblings of Psilocybe cubensis in Australia. The other thing you need to understand about SplitsTrees are trying to display all of the relationships among data, and complex relationships like this lots of reticulation or reticulation at the bottom of this Australian group is usually a sign of recombination, although there are other reasons to explain it, other ways to explain it. So this reticulation may mean that golden teacher has been hybridized with things like blue magnolia, Malabar, leucistic, and new strains have been produced from evolutionary innovation by crossing different strains. Okay, so that's it. That's cubes done, and you're just getting practice. So in your minds, have a little think about how a SplitsTree might look if Psilocybe subaeruginosa is different to these Northern Hemisphere taxa, things like Psilocybe cyanescens and Psilocybe azurescens. Probably, if you did the exercise and your homework, you would be thinking of something where there's a really long branch connecting Psilocybe azurescens or Psilocybe cyanescens, and Psilocybe subaeruginosa is totally different to them. If they're different species, we certainly wouldn't expect cyanescens and azurescens to cluster within or among Australian populations of Psilocybe subaeruginosa. Now we're going to get to a SplitsTree next slide after, and you're going to see some color. I just want to explain where that color came from. This is essentially a structure plot, and it's me trying to work out how many populations or how many groups are naturally present in that data. This is just from a small data set of about 7,000 differences among those genomes. So each vertical line is one individual haploid mushroom or haploid culture, and each horizontal line is adding a population. So if we started with, there's these numbers down here, like K numbers. If we started at K equals 1, there would only be one color. When K equals 2, we're adding a population. And I can tell you that the Queensland population comes out and everything else would be the same color. So we're gradually adding more and more potential groups to the data. And when we get to around k=8 or k=9, we start to get some nice solid lines and we don't have any breakup. At around beyond k=10, 11, 12, we start to get this real substructuring in these nice looking groups. So that is where the colors in the next slide come from. So here is a SplitsTree of Psilocybe subaeruginosa. And one of the first things you'll notice is that Psilocybe azurescens and Psilocybe cyanescens down here in the bottom right both cluster within or among Psilocybe subaeruginosa collected from Australia. So definitely not what we would expect for two taxa that you would consider different to subs, okay? The next thing that we'll see that I want to talk about, this I've always explained this terribly as well. So in this, similar to the cubes, we've got siblings. So ravensborn, this is all from one mushroom. This is 5 different siblings, haploid siblings. From the Bunya Mountains, I actually have about 3 or 4 different mushroom caps, but they appear as siblings. Kane has done a lot of collecting and he keeps his mushroom caps or piliae separate. So things from Kunyani are from individual mushrooms. So you can see that these are siblings. Ellendale, which was collected by Jan, are true siblings. So you're starting to get a picture that each of these little clusters of groups is from the same mushroom. So they're really, really closely related. And then they branch from a bit further down. So a lot more genetic diversity present beyond just in one genotype that's from one bed of mycelium that's making all these kind of really closely related siblings. One of my favorite things here is this Geelong and Clifton Hill. So 2 different parts of Victoria, one collected by Kane, one collected by Bill. Thank you to you both. And we see that they almost have a sibling relationship. Kane's definitely a siblings, Bill's probably not. And I believe this is from transporting mulch throughout Victoria or wood chips throughout Victoria. And you've got this genotype that's kind of invasive, and it's sort of shed some light on what might have happened to introduce Psilocybe azurescens or Psilocybe cyanescens to the Northern Hemisphere. We haven't sampled. If we kept sampling from New South Wales and maybe more from Eastern Victoria, we'd really expand this wagon wheel of this network. And probably we'd end up sampling from the original, if not the original, a really closely related genotype to these ones in the Northern Hemisphere. I think that's all I need to say about that network. This is relationships among the ITS region of those 86 genomes, maybe not all 86 made it in there, and then a whole bunch of ITS sequences I've just downloaded from GenBank. So this is a dichotomous hypothesis of evolution. It's not a SplitsTree. It's showing one potential hypothesis of sister relationships among all these little individuals. So the squares are from GenBank, the circles are our data. Now, the goal of these kind of, or applying a name, is to have a monophyletic group in taxonomy, which just means that if you've got Psilocybe subaeruginosa, you want it to all come from one most recent common ancestor, and everything from that most recent common ancestor is a sub. When you start breaking up a group with cyanescens or azurescens or weraroa or allenii, then it makes Psilocybe subaeruginosa paraphyletic in regard to those taxa. There's a few solutions. You could describe more and more and more to reflect intraspecific diversity of ITS, which is not a good solution, or you call everything Psilocybe subaeruginosa. You can see here that just from our SplitsTree before, we've got all these red dots, which is Victoria, South Australia, Tassie. Oh, I forgot to say, yeah, that population is kind of divided by a range. It's too late, I'll let it go. But you've also got ITS types from that same population in other parts of the tree. So ITS is intraspecifically variable. among Psilocybe subaeruginosa in Australia. It's a great marker to say, hey, this is in the subaeruginosa complex, or this is Psilocybe subaeruginosa. It's not the best marker to start dividing up Psilocybe subaeruginosa into new species. One thing that is interesting, and it's sort of hidden by this UQ brand here, is that this is Psilocybe makarorae. It's sister to the complex. I didn't really make a root. I just did a midpoint root, but it is different to everything else in there. It actually differs by 11 base pairs to this clade. So at some point in time, a taxonomist, definitely not me, will have to decide whether everything is subaeruginosa and makarorae is sister, or whether this is all subs. Okay, I just want to show another little piece of evidence about why subs are native, or the center of origin is in Australia. And I've always explained this terribly, so this time I've made a figure. Now, Basidiomycota, mushrooms, they have an inbuilt genetic mechanism to help them outbreed, and it's called tetrapolar mating. And it means that at two different independent spots in the genome, that control mate compatibility. And so the example that I've just made is on the left here, we've got one locus, the PR, which is the pheromone receptor locus, and it can either be a positive or a minus. And in a parent, you would have a positive and a minus allele, and each of its 4 little basidiospores would get one each of those. Sorry, we'd either get a positive or a negative. And so a positive, same thing with everything in mating, opposites attract. A positive can cross with a negative. So in bipolar mating, which is when you only have compatibility controlled at one locus, you can cross with 50% of your siblings. Tetrapolar mating, so easy. You're just introducing another locus. And in this case, it's the homeodomain locus, the HD locus. And really, it's the exact same thing. You've got a parent that has these positive minus at HD and PR genes, and the offspring each get this makeup, either a positive minus positive minus, and you can only cross when you differ at those loci. So in tetrapolar mating, you can only cross with a quarter of your siblings. Oh man, I hope that makes sense. So in cubes, that PR gene, the pheromone receptor locus, it's responsible for when hyphae meet, they can recognize each other. They go, hey, well, we're the same species and we differ. Let's get this on. Let's let our hyphae fuse, and we can start seeing what cooks from there. So in cubes, we resolved that this is just the Australian population. There's pretty much 3 alleles. This one's a little bit more complicated. There might be 3 alleles just within that one. So maybe there's 6 alleles at the PR locus. And we know that the Australian cube population is actually pretty diverse compared to the cultivated populations. In fact, the cultivated populations really only have 2 pheromone receptor alleles when you look across those 100 or so genomes. So let's take a look at what the diversity looks like in subs. And we've got heaps of alleles of the pheromone receptor locus. And it's because in the center of origin, you expect the most genetic diversity to occur. It's where things evolved. You want to be able to outcross, so you want to differ at this locus. So it's in its interest over time to differ at the locus. And we would expect the center of origin to have a lot of diversity here. In support of a hypothesis that everything that we've looked at is Psilocybe subaeruginosa, if we think back to our structure plot and our SplitsTree, the bunya clade up in Queensland was always the most genetically different to everything else. And just this afternoon, I've had a look at some crosses that I did from Queensland in the Bunya Mountains with things from Tasmania, with things from Victoria and South Australia. And you have all these compatible reactions. Black is a compatible reaction. Red is an incompatible reaction. Some of these red ones, I'm not sure, when you see a long red, it's a little bit of a surprise to me. I really was crossing things in the same with the same allele to make sure that they didn't cross. So it's always a surprise. I think I might have to go back and look at these a little bit more closely. But yeah, Australia has a rich diversity of mating alleles and things from the Bunya Mountains that are really genetically different to things in Tasmania can cross, which may support that Psilocybe subaeruginosa is one big species that has these disjunct populations or phenotypes that have been introduced to the Northern Hemisphere and then been described as new species. How long do I have? 10:20. Oh my gosh. I think I won't do justice to this. This structure plot is of cubes. Pretty much it really made me think about the difference ecologically between cubes and subs. So, and it's right here in this Talabudra. This is actually temporal sampling. Since I started in 2020, 2021, 2022, I've made collections, sequenced genomes, and over time, the same genotype doesn't occur. So the way that I think of cubes now is that they're shedding spores, cows eat them, goes through a cow, comes out in a nitrogen, carbon-rich environment. It colonizes manure, fruits, and that genotype is ephemeral. After that manure disappears or that mycelium disappears, that genotype is gone forever, but the next lot of spores is ready to get hoovered up by a bunch of cows or mammals eating grass. Subs are the opposite. Subs are forever. They're perennial. That genotype is persistent over time and space. It means that if you've gone to a sub patch in 2023, 2022, 2021, whenever, who knows how far back in time, you're visiting the same mushrooms. You're visiting an old friend that's the same genotype every time you visit it. So cool. So just the comparison of those two things is like an annual plant to a perennial plant, cubes and subs. Okay, the last thing I really want to talk about is just about the developments in woodlovers paralysis and looking at the psilocybin diversity, the diversity of the psilocybin locus in subaeruginosa. So just a little bit of background. Of course, tryptophan, an amino acid, is metabolized into psilocybin by these 4 genes, Psi-D, Psi-H, Psi-K, and Psi-M. That's in every magic mushroom, has this pathway. And then some other enzymes, Psi-P, removes a phosphate group from psilocybin to turn it into psilocin. Psilocin then polymerizes, it joins together to make that bluing reaction, and that's a reversible reaction. And you can imagine that along the way, if you've got more expressed Psi-D, you might end up having a higher concentration of tryptamine. Or when you start to change the function of these enzymes, things might start to happen. And that's kind of what we were looking at, I think, at the end of last year, and that I've shared with you before. So this is a gene map of the psilocybin locus, comparing our woodlovers, so subs, and then a cube at the top. So subs differ to cubes. Cubes have a gene inversion of Psi-T and Psi-K. Subs are going in whatever, the other direction. And then subs have 3 homologs of Psi-H. And in the genomes we looked in here, these 3, Psi-H3, this third gene was probably a pseudogene. It had a premature stop codon, but it was still annotated as a gene. So Psi-H is the gene that metabolizes tryptamine into 4-hydroxytryptamine. And when you have additional copies of a gene or an enzyme floating around and the function is already taken care of, it can innovate, it can start doing other things to compounds. And since I've been talking to some of my colleagues who are working on neurons and serotonin receptors, it's really got me thinking. So humans have those at least 14 serotonin receptors. And psilocin has an affinity to particular ones, but it can also bind to other receptors. And what I found interesting was that serotonin receptors occur in the peripheral nervous system, and they have been linked to things like the symptoms of Parkinson's disease, so shaking. And I just thought, wow, wouldn't it be amazing if an analog of 4-hydroxytryptamine or tryptamine was binding to a peripheral serotonin receptor to cause a paralysis? Anyway, let's have a look at what we were looking for. So In those 86 genomes, there was about 6 that had a functional copy of CyH3. And those are the 6 isolates that I will be looking at, sending to a chemist. I won't be doing it, to have a look at the metabolites that are being produced. So across those 86 genomes of Psilocybe subaeruginosa, there is a lot of diversity at the psilocybin locus. So it's kind of shaped by population. So something in the Bunya Mountains is producing psilocybin at or has genetic variation. That genetic variation could translate to different things, such as upregulation of one gene over another, better transcription. Who knows? It's just genetic innovation over time. So even though the end product is psilocin, maybe there's something else going on in the pathway based on differences, genetic differences. The 6 genomes that had woodlovers paralysis, I know that 5 of them are this little cluster here. So things that were collected in Clifton Hill by Kane and Bill in Geelong. And I'm not sure where the last one is, but yeah, super interesting. So some of those wood chips that Kane and Bill were looking in were potential woodlovers, in my opinion. And I'm going to leave it there. I'm sorry that it's been so rushed, but yeah, thanks, Emily. Good one.
Thank you very much, Alistair. And I told you, it's, kinky stuff about mushrooms.